Okay, so in this video, we're going to mainly focus on plasmid-based transformation and how scientists can isolate a particular gene of interest and then use bacteria or prokaryotes in order to make copies of it. So when we look at gene cloning, we're going to use a vector called plasmid. Now, plasmids are naturally occurring circles of DNA that are not part of the main chromosome of a prokaryote, but rather they're like extra pieces of DNA that can have anywhere from 50 to 500 genes on them. So scientists are going to use this plasmid for cloning. So now when we talk about how do we use these plasmids, scientists are going to make recombinant DNA. So with recombinant DNA, they're going to use a plasmid, and then they're going to take the DNA that they're interested in using to study. So for my example, we'll be using insulin as the human gene of interest. So, luckily for scientists, they discovered these cute little enzymes called restriction enzymes that they can use to cut DNA at a very specific sequence. So, with that, a restriction enzyme, they cut at, let me go ahead and move my face. So, restriction enzymes, they cut at a specific sequence, and anytime they see that sequence, they cut the DNA. So, when I say that they cut the DNA, what that means is they are going to find the restriction site to cut it at. So here, anytime this restriction enzyme, in this case it's EcoR1, anytime it comes across GAATTC, it's going to cut the bonds in the DNA molecule. So here it would break the phosphodiester bond, and then it would break the hydrogen bonds, and then again the phosphodiester bond. So when it does this, it produces little like tags that hang off of the new fragment of DNA that's been cut. And the cool thing about DNA is that it's universal. So A, T, C, and G are in every organism. So if you cut like human DNA with EcoR1 restriction enzyme and produce sticky ends AATT, well, if you cut a plasmid with the same exact restriction enzyme, anytime there's that sequence, that restriction enzyme will also cut the DNA there. Now, the cool thing here is that so where did these restriction enzymes come from in nature? It turns out they were discovered in prokaryotes. If you remember viruses, bacteriophage viruses insert their DNA into a bacterium in order to infect it, and then it'll go through the lysogenic or the lytic or lysogenic cycle. Well, bacteria, prokaryotes, they actually have restriction enzymes to cut up that virus DNA and therefore kind of protect themselves from being infected with phages. So anyway, in the 80s, scientists discovered these restriction enzymes and now have learned how to use them for our own advantage. So with these sticky ends, anytime A is near T, they can base pair together. So we'll take advantage of this base pairing rules in DNA. And now these sticky ends, what we can do is we can actually mix the plasmid with the DNA from a human, for example, with the fragments, and they'll pair together. So now we have what's called a recombinant chromosome or recombinant DNA. So this plasmid is made of DNA with from two different sources. And thanks to those complementary sticky ends, they were able to base pair together. So when we look at what scientists can do with this, so if we take the plasmids and they are outside of the bodies of a bacterium, out of the little cell, let's put it here. So what we could try to do is we could try to have that bacterium take up the plasmid, take it into its own self, and then express that gene. So here when I say express that gene, that means transcribe and translate. So here the RNA polymerase will do transcription and make messenger RNA, and then a ribosome can translate it and read it codon by codon and build like human insulin, for example. But one thing to be careful or be mindful of, when you want to have a prokaryote express human DNA, the thing is that in humans or in eukaryotes, what we do is we do RNA processing. So for our section of DNA, we have, when we transcribe it into RNA, we have what's called introns and exons. In prokaryotes, they do not have introns. There's no RNA processing. So you kind of want to make sure that the DNA you insert into the plasmid has already had the sections that code for introns removed. Okay, but here you can have prokaryotes that make lots of copies of like growth factor or insulin for us. Okay, so there's different kinds of restriction enzymes that are each going to have their own sequence to cut. So if you are going to make recombinant DNA, you want to be careful that you use the same restriction enzymes if you want the base pairs or the sticky ends to match. Kind of like craft scissors. They each have their own sequence. Okay, so now here you have some plasmids, and some of these plasmids have your gene of interest and some of them don't. Also, when we mix plasmids with prokaryotes, sometimes you can't see inside to see which one actually was successful at transformation or taking up the plasmid. So when you mix prokaryotes and plasmids together in an environment, not all of them are going to do transformation, and then even then, not all of the plasmids will have been successful at forming recombinant DNA. So, what do we do about this, right? Oh, sorry. Okay, so, sorry, I should have deleted these slides. So now let's think about this. What can we do? Like, how do we know if a cell is actually successful at transformation? Well, the plasmids that we start with, before we even attempt transformation, we're going to use genetically engineered plasmids that are going to have a section on them that code for antibiotic resistance. So the plasmid to start, before you cut it with the restriction enzyme and have the gene of interest inserted, you're going to start with one that already has a gene for antibiotic resistance. So why this is important is because if you have a plasmid with a gene for antibiotic resistance and you make a petri dish or a nutrient agar, if you pour the plates with antibiotics already part of the growth medium, now only bacteria that have that gene for antibiotic resistance will survive. So the bacteria that have taken in the plasmid with antibiotic resistance will be the only ones to actually survive and grow on that nutrient medium. So here, if these little plasmids, the red line represents the gene for antibiotic resistance, when you mix the plasmids with the bacteria and you're not sure which ones were successful in transformation, here, if these bacteria are exposed to antibiotics, then that one dies, and you know only the one surviving took up the plasmid. So let's go ahead and see this. So here, if you make the petri dish with with antibiotics in the agar, any bacteria that did not have successful transformation, they did not take up your plasmid, they don't survive. So now all of the colonies remaining on that nutrient medium will have the plasmid and have successful transformation. Okay, so the gene for antibiotic resistance is really important to help you isolate which bacterial colonies actually had successful transformation. Okay, so sometimes when you look at a picture of a plasmid, for example, it looks something like this. So maybe the green arrow is the gene that you're interested in studying. Maybe it's the section that codes for insulin. And then you have your promoter sequence. That's going to be where RNA polymerase attaches to start transcription. And then that blue section is the section that codes for antibiotic resistance. So if for some reason this section is disrupted, well then this plasmid would no longer confer resistance or give resistance. Now this orange section here is the origin of replication. This is where when this plasmid is copied or goes through DNA replication with helicase and DNA polymerase and ligase, etc., it's going to start here. So that's just the origin of where replication starts. It's not really involved in working with your gene of interest. Okay, now sometimes you can have restriction sites that are located within important genes, and that's how you can identify as well if you had your gene of interest inserted or not. Okay, so that is plasmid-based transformation.